Hello, good afternoon. My name is Josh Jackman, and I'm an assistant professor in the School of Chemical Engineering at Sungkyunkwan University in South Korea. First of all, I'd like to thank the conference organizers for giving me the opportunity to give this talk today. I will be presenting on broad spectrum antiviral strategies for stopping membrane-enveloped viruses. So as we've seen, especially this year, more than ever, viruses are one of the biggest global health problems in the world. This year, we've seen the COVID-19 pandemic, which is still ongoing, and it's had a huge effect on the global society. But this is not the only virus. There are many different types of viruses that affect our daily lives. Millions of people live with chronic viral infections, such as hepatitis C and HIV. Many others have respiratory viral infections every year, such as the influenza virus. And now there's also concerns about new types of coronaviruses, such as COVID-19, potentially becoming endemic, problems that do not go away easily. So there's many different types of viruses that are global health problems, and there's a lot of attention to developing new medical strategies to stop viruses. So when we think about viruses fundamentally are parasites. By themselves, they can't do much. They can't replicate by themselves. Instead, they need to infect living cells, such as human cells. And what they do is viruses infect and enter human cells, hijack the different parts of the human cell, use those parts like machinery to produce new virus copies. Those virus copies are then spread to infect other cells. So how do we stop this parasite? One way that researchers have tried to develop antiviral medicines to stop viruses is to stop them from replicating and producing new viruses inside infected cells. And there's interest in these kind of approaches because there's many different viruses use the same machinery inside infected cells to replicate. So if we can develop effective antiviral strategy to stop these production processes, we can potentially stop many different types of viruses. However, depending on the type of infection, it can be challenging to develop an effective antiviral strategy based on these replication inhibitors because they only work once the virus has already infected the cell. This has led researchers to also develop what we call entry inhibitors, which are antiviral strategies that directly inhibit virus particles and stop them from entering cells in the first place. If we stop the virus particle, we can prevent infection and we can actually stop the process from starting in the first place. So there's a lot of interest in developing these entry inhibitors that directly act against virus particles, and a lot of interest you might see in the newspaper these days in terms of antibody therapeutics and different things that specifically bind to COVID-19 virus particles, for example. But the challenge with these entry inhibitors is that they're more specific to one type of virus, which is good when we know what the virus problem is, but it's difficult to develop very specific medicines for every type of possible viral threat because there's so many. So current entry inhibitors typically work against only one type of virus each, but we want to develop new strategies to create entry inhibitors that work against many different types of viruses, both today's threats and tomorrow's future potential virus problems. So now, as we're in the middle of the COVID-19 pandemic, our first priority is to stop the current pandemic and to go back to normal life. But already many researchers are already asking, how can we stop the next pandemic? This image is from a New York Times article earlier this year, and there's many people in the policy communities, biodefense communities, academic research communities, pharmaceutical company communities that are interested in these questions from many perspectives. You know, COVID-19 was a major challenge to global health infrastructure. Even if we now see in the newspaper in the past week or two that vaccines are being developed, it'll take a long time to actually get them out and reach a large segment of the population. So what people are now thinking, how can we preemptively stop the next pandemic more quickly? What can we do better? There were certain positives from the current response to COVID-19. There were certain areas for big rooms of improvement. And fundamentally, people are now asking, how can we be better prepared for the next pandemic? And one of the key questions related to this pandemic preparedness is, what will be the next virus? There's so many different viruses circulating in nature among animal populations, among human populations. What will be the next virus that causes a major outbreak? This is a key question. It's the trillion-dollar question, literally. But it's not easy to actually pinpoint one virus. There's many high-risk viruses that have certain properties that make them threats to cause future pandemics, but we don't know exactly which one. So we need to be ready for the unknown while recognizing that if we put all our resources into developing a strategy to stop one particular type of virus, this might not necessarily be the best investment. We need to develop strategies that have the potential to work against many different types of viruses. And this creates the concept of broad spectrum antivirals, developing antiviral strategies that can work against multiple types of viruses, including ones that could potentially cause future pandemics. So recently, our research group has been thinking about this question in terms of virus structure. So what we did is we actually asked a similar type of question to pretty much everyone else listening to this talk and in the scientific community at large. What type of virus will cause the next pandemic? And to kind of begin thinking about this question, what we did is that we created a kind of summary of representative virus epidemics and pandemics that occurred during the past decade, 2010 to 2020. And we looked at aspects like, what was the virus? What years did it occur? Where did it occur? What was the virus particle structure? And how many infections and deaths did it cause? And one thing here that we did, which is a bit unique compared to most kind of summary studies, is that we really emphasize this virus particle structure. So when you think about virus particles, there's two main types. Some virus particles have a lipid membrane envelope, and this lipid membrane is necessary for the virus particle to be infectious. And other types of virus particles are non-enveloped. They usually have a protein coating on their surrounding, but they do not have a lipid membrane. And what we saw surprisingly was actually over the last decade, nearly all virus epidemics and pandemics were caused by membrane-enveloped viruses. We saw many recurring themes, such as mosquito-borne viruses, such as dengue, yellow fever, chikungunya, cause many outbreaks, as well as coronaviruses, such as MERS and COVID-19, along with other very serious viral threats, such as highly lethal influenza strains, Nipah virus, and Ebola virus. And when we thought about this together, based on the analysis, it becomes quite clear that enveloped viruses are a major threat, and we need to do more to stop them. So we thought about, how can we actually develop focused strategies to stop membrane enveloped viruses? In this page, this is just an overview of different types of lipid membrane envelope viruses. In A and B, we see dengue particles. C through J, we see influenza virus particles budding from cells. K through L are COVID-19 particles. And M is African swine fever. And what all these diverse viruses, some infect humans, some infect animal livestock, what all these viruses have in common is that they've caused major viral outbreaks that have huge health and economic consequences, and they all have a common feature, a common structural feature called the lipid membrane envelope. This lipid membrane surrounds the virus particle, is necessary for structure and necessary for infectivity. So if we can stop this viral membrane, we can stop the virus. So our group and many other groups worldwide have been thinking about how can we effectively stop the viral membrane, essentially disrupt the viral membrane or prevent it from functioning. And this problem is very challenging because the viral membrane surrounding envelope viruses is actually derived from human cell membranes or host cell membranes in a more general sense. So the lipid molecules in the envelope virus membrane are similar to human cell membranes. So how can we develop specific strategies to target the envelope virus while not damaging human cells? This is a very big question and one that we and many research groups have been thinking about critically. In terms of different types of envelope virus inhibition strategies, some researchers have developed strategies to bind specific antibodies or small molecules to certain lipids in the viral membrane and stop it from functioning. Other researchers have developed small molecules that change the fluidity of membranes, viral membranes, to stop them from fusing and infecting human cells. We and other researchers have developed selective strategies to form small holes called pores inside viral membranes so that we can selectively disrupt virus particles but not damage human cells. In order to develop these selective antiviral strategies to damage virus particles and to inhibit virus particles but not damage human cells, we've found great potential in using engineering technologies to actually study the mechanism of action of antiviral drug candidates to create high potency and selective antivirals that work against envelope virus particles. So using biosensor technologies such as quartz crystal microbalance dissipation, we can create artificial cell membranes that mimic envelope virus particles and actually study how compounds such as antiviral peptides selectively form pores in these virus mimicking membranes, and how we can exploit mechanisms such as membrane curvature sensitive pore formation in order to create highly effective antiviral strategies. We've also seen great potential in other types of engineering technologies, such as nanoplasmonic sensors, which have allowed us to really deeply understand how certain types of antiviral peptides can be selective for membrane curvature. And this allows us to develop antivirals that work selectively against envelope virus particles with very high membrane curvature, and also to understand why these peptides do not work against low curvature human cell membranes, which is a very important distinction. Creating a drug that selectively targets high curvature viral membranes, but does not affect low curvature human cell membranes. By using engineering technologies, we've been able to do this. We've also been able to use various types of microscopy techniques, including time-lapse fluorescence microscopy imaging, to directly look at the interactions between individual virus particle mimicking membrane structures and antiviral peptides in order to do highly parallel imaging on hundreds of these individual particle interactions at a time. And this allows us to look at the effects of peptide concentration, membrane curvature sensing, and so forth to really optimize drug development and develop the best antiviral peptides. So based on these engineering technologies, we've been able to develop a new concept called lipid-envelope antiviral disruption, or LEAD is the acronym, in collaboration with the World Health Organization, National Institutes of Health, US Army Medical Research Institute of Infectious Diseases, and Public Health England. So we've been able to develop this LEAD concept to actually develop antiviral peptides that selectively inhibit membrane-enveloped viruses. In this LEAD concept, we've explored in terms of various classes of mosquito-borne viruses. We've seen that it works in vitro and in many different in vivo models against viruses such as dengue and Zika. And we're also continuing to explore other type of mosquito-borne viruses, including yellow fever, chikungunya, West Nile. And one of our key focuses has been Zika virus, which is a virus that causes infection in brain. And we've actually been able to develop a lead concept antiviral peptide called the AHD peptide, which is able to inhibit envelope viruses, and it's able to cross the blood-brain barrier in order to treat virus infections in the brain. And in a mouse model of lethal Zika virus infection, we saw that this strategy could decrease viral loads, prevent brain damage, reduce clinical symptoms, and also protect against lethal infection. So one of the most exciting properties of this AHD peptide was that we could start the therapy as late as 3 days after the mouse was infected with the virus, and we still saw very good clinically relevant results. So when we infected mice with a lethal dose of a Brazilian Zika virus isolate from clinical patient, we saw that when treatment started 3 days after virus infection, we saw that we could still protect the mice with very high protection against death. So 10 out of 12 of the treated mice survived, whereas all the mice in the untreated group died. And the therapy also prevented weight loss, protected against eye pressure increase, and protected against high white blood cell count, which are all typical indicators of Zika virus infection in mice. And we were also able to see that the AHD peptide could penetrate the blood-brain barrier and inhibit viral infection in the brain. So when the peptide was systemically administered in the bloodstream, it could actually cross the blood-brain barrier and directly inhibit viral infection in the brain. This is really amazing because the blood-brain barrier was still intact and it could actually cross intact blood-brain barrier. For most class of antiviral compounds, such as antibody therapeutics, it's very, very challenging to cross the blood-brain barrier. So this was a huge accomplishment. And we saw that the antiviral peptide could also prevent the loss of blood-brain barrier integrity by mitigating the viral infection in the brain. So we've also seen that this same peptide can work to prevent transmission of Zika viruses to fetuses in a pregnancy mouse model involving pregnant mice. And we could determine this in terms of mitigation of Zika virus in the brain tissue of the fetuses, along with improved fetus viability and improved fetus weight. Now, the Zika virus is a very prominent example of a recent human virus threat, and we're continuing to explore also against COVID-19 and many other types of viruses. But I also want to turn your attention to a virus that has had a huge impact globally in the last 2 years, and which you maybe have never even heard of. And this is a virus that is wiping out the pig population in a large part of the world, especially in Asia, and it's called the African swine fever virus. This is creating a huge impact on food and feed markets. And I just wanted to also introduce you to this other type of envelope viruses, which is having a enormous effect on the agriculture sector, and is also another type of enveloped virus that we need to stop. So over the past few years, since around 2018, there's been major outbreak of the African swine fever virus in Asia, and in addition to Eastern Europe and the Caucasus region, but especially in Asia and parts of China. The ASF virus has been a huge challenge, and it's resulted in a large fraction of pigs in China having to be culled or killed due to stopping this virus. So it's been estimated that at least 30% and approximately up to around 50% of pigs in China have been killed over the last 2 to 3 years due to this ASF virus. And it's having a huge consequence for pork prices worldwide. So it's led to pork price increase and scarcity. And there's major concern about this virus spreading and damaging pig production in other countries worldwide, including in the US. US government and USDA and FDA in the US has highlighted that African swine fever is one of the major threats that could severely disrupt the US agricultural economy. So in this, for these reasons, there's been a lot of interest in understanding how this ASF virus transmits and how we can stop it. So it's been discovered that animal food called feed is one of the main transmission vectors. And there's been a lot of interest in developing antiviral strategies, including feed additives that can be placed in the feed in order to stop the virus. So recent work by Kansas State last year, 2019, established that ASF can be transmitted by liquid or feed, and additional epidemiological studies have pinpointed that ASF was likely transmitted by feed in past outbreaks. So there's enormous attention placed on developing antiviral feed additives that can actually inhibit ASF in the feed and stop transmission. So until recently, one of the most common feed additives, chemical feed additives, that was used to stop bacterial infections and also potentially mitigate against viruses was called formaldehyde. Now, formaldehyde is still used in certain parts of the world, but many different parts of the world, including the European Union, have begun to ban formaldehyde due to negative health effects, such as increased cancer risk, because this molecule is carcinogenic. So there's been a lot of interest in exploring natural feed additives that can replace formaldehyde. And one of the main types of compounds that my research group, in collaboration with experts in the United States and in Singapore, have been looking at are medium chain fatty acids and monoglycerides as feed additives for pig production, including looking at aspects such as how we can improve gut health performance, as well as mitigate feed pathogens such as ASF in the animal feed. So there's various types of saturated fatty acids and monoglycerides that are very important. And on the bottom of the screen, I'd like to point your attention to one in particular that we're very interested in, which is glycerol monolaurate, or also called monolaurin. It's one of the most potent antiviral compounds in this class, and it has enormous potential in terms of its membrane disruptive activities against envelope viruses and bacteria. And we think there's a lot of interest in using this type of compound as a potential feed additive. So we've recently published some work in the agricultural journals discussing fatty acids and monoglycerides as virus-killing feed additives, how they could actually potentially replace formaldehyde and really provide a superior advantage to kill virus particles in the feed, as well as potentially stop bacterial cells, inhibit bacterial contamination as well. So really, this is kind of a 1-2 punch multifunctional additive that's really based on the lead concept. It's a practical example of the lead concept for stopping a membrane-enveloped virus of agricultural importance. So we've been using engineering technologies, these same biosensing technologies that we've used for studying antiviral peptides. We've now applied them to explore antiviral feed additives based on monoglycerides. And this has allowed us to kind of pick the best monoglycerides with the best performance, optimize their dosing, composition in order to get the most potent membrane disruptive effects. One of the advantages of these type of compounds is that they're membrane active, so that they can disrupt membrane enveloped bacteria, as well as disrupt membrane enveloped viruses. So for agricultural community point of view, this is a huge advantage to stopping both. And if these type of additives work effectively, we no longer need either antibiotics, which are being phased out, and we also don't need to use formaldehyde anymore. So in preliminary studies in vitro experiments, we've seen that GML had high antiviral activity against the African swine fever in liquid conditions. It inhibited around almost 99% of the virus. And we also saw some performance at 5 millimolar concentrations of other types of fatty acids, especially capric acid. However, at 250 micromolar test compound concentrations, only GML was still active, and we still saw a nearly 99% drop in viral infectivity. So it really led us to conclude that GML is a very promising compound to inhibit African swine fever. We've also continued exploring GML in a cocktail of medium chain fatty acids to actually inhibit ASF directly in feed. And we saw that when we doped feed with these compounds and then added virus, we saw that the GML significantly inhibited virus in the feed within time periods as short as 30 minutes. It was also effective over longer periods, and also demonstrated much higher efficacy than the fatty acid combination tested in parallel. So this was very kind of promising initial evidence that monoglycerides such as GML can reduce ASF in animal feed. And this just is a promising example more generally of how we can use engineering technology in combination microbiology testing to develop really high performance agricultural innovations. So normally when we think of engineering and biosensors, we might think about how these technologies can be used for human medicine, and they certainly can be, and they certainly should, and there's huge potential there. But also we want to kind of expand the viewpoint to see how engineering technology can be useful for human and agricultural applications. In general, we think there's a lot of potential to continue exploring how engineering technology can develop new strategies to stop membrane-enveloped viruses. So I'd just like to conclude my talk by thanking my collaborators and colleagues who are involved in this research, as well as the grant agencies and other organizations that have supported this research. And thank you very much for listening to my talk. I'm sorry that I couldn't join you today in Singapore, but I hope that you have a great rest of the conference, and I look forward to seeing you in the future at future conferences. Thank you very much.